Thank you very much for the invitation, and I will tell you two stories or about two techniques that I worked on in my PhD, in my postdoc, and now in my own lab in the IMP in Vienna. So starting with the work that I did in Holger Stark's lab a couple of years ago together with my colleagues there, is on protein stability of complexes by thermofluor screens. So the question is it actually a trivial process to go from to get your optimal sample? And also from the talks in the morning you saw, it's not really trivial. You cannot just put a sample on a grid and assume that you get a perfect, beautiful grid. Often you see a lot of aggregates, you see a lot of broken particles, and only a few times you really see what you want to see. The big question, however, is how do you get from a sample that is basically fallen apart to a sample that is beautifully intact and where you can get a structure of. And ideally, you want to do an optimization that consumes a little amounts of sample, that is highly sensitive, that you can do in high throughput, and it doesn't need special instrumentation that you still have to buy. Luckily, there is a method, and we heard of that in the morning, that fulfills all of this, and this is thermofluor or DSF. So we just need a few micrograms of your sample. It can be completely automatized, and the instrumentation is mostly a real-time PCR machine, which you have. And we use a gel stain, which is Cypro Orange, which is cheap and everybody can buy. The problem with this is, and we heard that already, that not always the data is really interpretable, especially not for protein complexes. So literature claimed, when I started the project, literature claimed thermofluor is not applicable for multi-domain proteins or protein complexes at all. We took the challenge and actually tried it, and Ashwin in the lab actually showed already that it was possible, and we just had to come up why. So how does it work? And I'm just going to be quick. We saw that already. We have our folded protein in a solution that has our dye, and as soon as we heat it up, the sample, we will see that at some point the protein will unfold, it will bind the dye gets hyperfluorescent, we get signal, and as soon as we heat up even more, the unfolded proteins will start aggregate, release the dye again, and we get a dropping signal. So ideally, we have a single domain protein, and then the curves look like that. Then we have a very steep transition, and from this we can get the melting temperature, and this melting temperature will be useful to analyze the data. However, often when we have complexes in not ideal conditions, we get multiple transitions, and these multiple transitions have different melting temperatures. So what is the melting temperature that we're interested in? What is the right melting temperature? And sometimes, if these transitions are very close to each other, we get curves that look like single transitions, but they aren't. They're just overlaps of these individual transitions. They have higher melting temperatures, but they're not more stable. And this is why literature claimed that. Literature saw these curves, and they could sort their curves just by the TMs, and they got meaningless results. So they couldn't really interpret these results. However, you also get for protein complexes this kind of curves. This one-step unfolding is two-state unfolding, and this is stable curves. And the only thing we had to come up with, we had to get a biophysical model that described all of them and then give us parameters that are actually meaningful that we can use to sort these curves and tell you what is a stabilizing condition and what's not. So what I came up with is a formula that is slightly exhaustive, but you don't have to remember it. We published it so you can look it up. But quickly to tell you how we came up with this and how we come to that. So for the single domain protein, you can look it up in textbooks. That's quite easy. But what happens for complexes? So the two extreme cases of what can happen to a complex when you heat it up, well, it can first disassemble, and then the individual components can unfold. Other extreme case, everything can happen completely cooperatively. Everything unfolds at the same time, and you get a fully unfolded protein cooperatively. And any other case is somewhere in between. So our formula is able to actually do that by modeling the individual parts of these individual parts by just having the cooperativity in a constant, and we can actually fit whatever curve. So we can, with this formula, we can fit every experiment that we see, almost every, unless there's huge artifacts. And then we get a number of physical parameters that we can extract from this fitting procedure, which is the real, or at least something close to the enthalpy, and the apparent melting temperature, which is not the melting temperature that you just get from taking the derivative of your system. And what do we do then? This is a two-step process. We will find the curves that fulfill a two-state model, and only these curves we can really analyze, where we can use melting temperatures. So where the fit is valid. So we first look, is the fit valid? If this is not true, we discard the curve because we know this is not a stabilizing condition. We cannot sort the non-stabilizing conditions, but we're not interested in like how unstable the protein is if we know it's unstable. Then, and this is the important part, is do we have a high delta H, and the delta H is the steepness of the curve. And only if this is sufficiently high and matches to the protein properties, we use the curve. Because otherwise, we have this overlapping curves and get TMs that are wrong. And in the very end, if all of this is true, then we can use the TM, and then we get meaningful values for that. We can get additional information from the curves and from the models, so we will see air bubbles quite nicely. We will be able to see nucleic acids, and we can get information from the cooperativity of aggregation. So this is not quite there that we can get meaningful information, but we can get some information out of that. And I'm showing you a couple of results. So to test all that and make sure that the curves that, how do we know that the curves that our model predicts are good curves are actually good protein, good stable protein? So what we did, and this is a method that we haven't heard of yet, but is a very useful method for quality control of protein complexes, negative staining EM. So we checked all the different conditions in negative staining EM, and you see, and for this protein, which is called cell A, and we have this condition here, we see large aggregates, not really good. If we change the pH a little bit, it looks much better. We get individual particles, but we still get these smaller aggregates, the street-like aggregates, which tells us often that we're close to a good condition, but not quite there. And finally, in very good conditions, we get nice individual particles in different views even, from which we can get structures, and we actually get this cooperative unfolding. Okay, can we make an experiment where we can predict the outcome and actually see things? So this is ribosomes, and it's known as soon as we remove the magnesium from the ribosomes, we can get them separated into their individual subunits, and they will not be stable anymore as ATS ribosomes. And we can actually take the EDTA away, add magnesium back, and get it back, and we see that in the curve. We go from the same sample, we go from unfolding before taking away the magnesium, after taking away the magnesium, and adding it back the magnesium, and we get a nice sample where we can calculate a structure. This is just a placeholder, more or less here. Obviously, we can make higher resolution structures of ribosomes. And the last experiment that we did is, what can we do for samples where we don't know anything about? So we were approached by Jürgen Markel from the University of Mainz, who gave us a sample that was supposed to give us a structure, and you'll probably hardly see, but this will never ever give us a structure. So the sample is really like broken apart, and you see individual small particles, if you see it. However, what you will appreciate, this curve is terrible, so we can't really interpret that in any way, and even our fitting algorithm has problems with that. So we run the full screen, and we buffered in the hit. So we didn't repurify, we just rebuffered. And we see it actually looks already better, so it's not that harshly fallen apart, it's still not amazing, through rebuffering, but it already got better. And and if we look at the curve looks much better already because it seems like it's extremely stable. And if we actually purify the protein in that buffer, so we start all over, we use that buffer, just purify in that buffer, we actually get nice complexes where we can get structures off. And actually in this buffer conditions, we even get crystals. So you might appreciate them here somehow. But this is actually nice crystals. This is a hemoglobin from a snail, so that's why the crystals are red. Okay? What else can we do with that? Can we only look for stability, or can we also screen for small molecule interactors? And we use the system of GORIEL-GORIES, where the two proteins are separated, and once they're separated, we expose a hydrophobic cavity, and this hydrophobic cavity is bound by the dye. That's why we have the starting point here. As soon as we put not quite the right nucleotide in, the two proteins come together, which is known. But as soon as we add the right nucleotide, ATP in this case, we even get a thermal shift, and we get this perfect curve, and we can screen for lots of different ligands. Can we do it even more complicated? So can we use protein ligands? And yes, we actually can. And this is the anaphase promoting complex that Peggy talked earlier. And there we added a protein inhibitor to the system. We knew that it was an inhibitor. And as soon as we add this protein inhibitor to the system, you see the curve shifts. It gets more stable. And we can actually screen that. Lastly, can we do it even more complicated? Can we do a reconstitution experiment? So can we use, and this is now the pyruvate dehydrogenase complex that is made up of 3 components, and now make a plate where we just iterate in different ratios through the components and see what is the best ratio to mix them together to actually get a reconstituted complex. We chose this condition, which looked the best and actually gave a good graph. And we reconstituted and put that on a gradient, and we actually get a complex that behaves quite nicely on a sucrose gradient and gives a very nice curve. So we can actually use that for reconstituting the sample. So with the same method. At the end of my PhD, we looked at 84 different complexes that we got from collaborators all over the world, and we optimized their samples in all kinds of different biologies, different biochemistries, different amount of change, different masses. By now, there's probably more than 200 complexes looked at, and in most cases, we can improve at least a little bit. In some cases, the complexes are already quite good that came to us. Interestingly, with all these complexes that we have, we can do some statistics and actually look what is the pH optimum. So if we look in the if we look in the PDB and look what is the pH optimum for these proteins, because they crystallize in this pH, we see a relatively broad distribution, which, however, also has to do with the preparation of crystallization. If we look in the EMDB, so look at all EM structures, it seems like EM people are just guessing. So they just take one buffer, and this is basically 7.5. And this is just a guess and not really the optimal condition we see from our data. And this is our data. In our data, we see actually even that 7.5 is not at all optimal. It's some proteins like it a little more acidic, some more alkaline, but 7.5 doesn't seem to be really a hit in terms of the stability that we measure. So it makes actually sense to screen, and also for EM people, it makes a lot of sense to screen, because none of the samples that we looked at was actually stable at 7.5, which is opposed to physiological pH. So we made a patent, we commercialized the system, and made a machine out of that you can actually buy. And this is mostly for reproducibility, because pipetting, real good pipetting, is one of the prerequisites that you get good results. Because pipetting errors by students not carefully pipetting is the major source of error in this method. And we actually tested that and gave it to a couple of students in the lab. And the variability for the average PhD student is so high that we can get completely different results, so we needed to rely on pipetting. Robots. Okay, that's part one. So how do we find stabilizing conditions? This is still my work from before I had my own lab, and now that I have my own lab, we also are still interested in protein heterogeneity and how to optimize our samples. And last year, around that time, I met Philip Kokura from Oxford. He gave a talk in the institute, and I had a one-on-one with him. After his talk, I was so flashed that I thought his method is the best method in the world, and we really, really have to do something with that. My enthusiasm also flashed him quite a bit, because apparently he's a physicist, and most of physics people didn't really appreciate his method at all, and didn't find it really useful. So he was really appreciative that I find a lot of use for it. So we teamed up, and there is a PhD student from my lab and a PhD student that I pay partially because it's shared with another lab, Katharina and Tanya. With a postdoc of Philip's lab, we teamed up. I sent the two of them to Oxford for a couple of months. And we tried to make use of the method. So, but what is the method? So he uses interferometric scattering microscopy. So it's a scattering method, but microscopy. So we make images. The problem is if you make scattering microscopy of molecules, we're diffraction limited. So our spots that we will see are naturally limited by the wavelength of light, which is 200 nanometers. So no matter whether we have a 2 nanometer protein or a 20 nanometer protein, it will always give us a diffraction spot of 200 nanometers. However, and this we also saw earlier in the morning, this signal and the intensity of the interferometric signal correlates perfectly with the mass. And this is what Philip's lab has done. So they have, and this is actually the signal that you get. So this is the molecules in the interferometric microscope. How is the molecules landing on the glass slide and going away? And from the intensity of these spots, we can actually determine the mass. We can calibrate that quite nicely with proteins that we know the mass of, and we get a 2% of mass accuracy. The detection limit is around 50 kilodaltons. In reality, in real samples that are not pure, highly purified samples, we rather are at 100 or 200 kilodaltons. And this is the data that we get. How is the experiment done? It's relatively easy. So this is a student in Philip's lab doing the experiment in real time. So this is the machine. We just opened it. And it's pretty much like a nano drop. We just pipette the fluid solution on the sample carrier here. We close the machine, switch on the light, and we immediately see our molecules coming. And now it's just a matter of how much statistics you want. Do you want like hundreds or thousands of molecules? The longer you measure, the more signal you get, and the better you actually can measure the mass of them. And we can measure the mass quite nicely, but what is this useful for? So what I was interested in is natural heterogeneity of complexes, and we look a lot on proteasomes and ribosomes. And when we purify proteasomes, we get like the normal bend pattern of the proteasome, but we get some additional proteins in there. And we're interested in these additional proteins, and depending from what tissue we actually get them, these patterns change. Now, the first question is, are these real complexes? Do they belong together, or are they just co-purifying? Where we have to do a lot of characterization and lots of biochemistry to actually figure that out. And in another example, we look at ribosomes that we get a structure of that have a couple of factors bound. However, the question, are they bound at the same time, at the same particles, or are they bound on different particles, and just through the process of averaging, we see them there. And this is a question we hardly can address with other methods, so we wanted to look at that. However, in many cases, flexibility is really a pain, so we wanted to exclude that flexibility will make much of an issue. So we went to the most flexible protein that we could think of that we got on the campus, and it was cohesin. And this is now, and you hardly can see that, but this is rotary shuttering EM, and you see cohesin is these ring-shaped molecules, and the ring can close and open and can make all kinds of floppy structures, which makes it very hard for Sec-Mals, for Sykes exclusion, for anything. It will always have really broad peaks because of this conformation of flexibility. In mass photometry, it turns out we get one peak, and this peak is exactly the mass that we expect. So this is really cool. So we can do things with that. But can we actually monitor also purification and optimize our purification? And this is again the interface promoting complex from Nick Brown's lab, and here you see there's a couple of fallen apart particles and that's not quite optimal. So we looked through the purification procedure that had an affinity step, as Peggy told you, an ion exchange and a size exclusion step. And if we look at any of these steps at the fractions that are good on the gel, you will see they look perfectly almost identical. So you have a hard time to actually distinguish them saying this is a good complex or this is not a good complex. So what we normally would do is we would take these fractions, look at negative stain, even sometimes make class averages and figure out, okay, where is actually the good fractions that we want to work with? So we did that in parallel. We did it with negative stain and made class averages and counted the particles that were intact and the particles that were not intact. This took my student about 2 days, while this took Philip's postdoc about 20 minutes. So in 20 minutes we could get what is the distribution of the different subcomplexes in each fraction of the signal, and we could actually figure out in what step and which fractions are the ones that we want to look at and actually get a nice, fully intact complex that we can do structural biology with it. I told you before that when we go to natural sources, there's natural variability in these complexes. So some of the complexes have some accessory proteins, some don't. We're interested in how many have these accessory proteins, does it make sense, and can we use strategies to enrich them? And we look at the proteasome because that's what my lab is looking at mostly, and you see here this is a purification from HeLa cells. And in this purification from HeLa cells, we see 2 additional proteins, and we wanted to know how much abundant they are. And we have here ECM29, which is one of the proteins, which is chaperone for the proteasome that we're interested in. And what is the different species of the proteasome? When you purify proteasome, you often have a hard time to actually separate the individual species because they are in equilibrium with each other. So we wanted to see what we can do. Can we actually enrich this or not? And for this, we made a salt screen. And we did it with mass photometry because this was really fast. So this whole experiment took Katharina like an hour. With data analysis, and we could figure out that the species that we wanted, which is actually this peak, is salt labile. Right? So the more salt we added, the proteasome gets loses this binding partner, though we actually showed that it's actually bound. And the mass shift that we see here is perfectly the predicted mass shift for ECM29 binding. What about nucleotides? Can we see, and this is well studied in literature, and there's more than 10 papers looking at the nucleotide dependence of the proteasome. And what we see basically is if we use different nucleotide states, we can perfectly recapitulate these experiments that again probably took several weeks, and this again was an experiment that Katharina did in an hour. So we can do optimization. At the moment it's not quite high throughput, so an experiment takes a minute, right? It's good, but it's not 96 well, will take 96 minutes, probably a little bit more with data analysis. But if you can parallelize that, and we hope that Philip is actually working on that, we can parallelize that and actually know which kind of subcomplexes are in our sample and how heterogeneous is the sample. So do you actually really want to go with something like that, or is that too heterogeneous? Or do you want to, when you want to enrich a certain species, this is the fastest way to actually do that. So many people are interested in membrane proteins, and Philip actually asked me once we did all these experiments, what do you really need to convince lots of biochemists that this is a useful method? And I told them, yeah, let's do membrane proteins, because I hear from many people that basically the devil made membrane proteins, because you can hardly work with them, and structural biology with membrane proteins is a pain. So we went to another collaborator that had a problem with a membrane protein, which is respiratory chain complex 1, which we got from Thorsten Friedrich in Freiburg. And here the problem that the E. coli complex is falling apart quite a bit. into subcomplexes. This is known from literature, but he didn't really know how to optimize that and actually get the full complex, because it's the only thing that he was interested in. And here you might see, so this is the negative stain image of the individual subcomplexes that we nicely see. And here you see the mass photometry signal. So you can see this signal is much stronger for the larger complex. So just that you see the contrast. So what can we do actually to optimize that? And Holger is probably telling you more about that. We can use cross-linking, and we do gradient cross-linking in a gradient containing the detergent element G, which is very low coming off the molecule, and containing glutaraldehyde in low concentrations. And with this, we can easily enrich for the species that we want, and we go really high in terms of fractions. So this is an optimization experiment that takes about 2 days. The actual mass photometry experiment took 2 minutes, maybe 5. And the last thing that I want to tell you, what can you do with it and what is the potential of it? Obviously, you can also measure KDs in one shot, and we did that on antibodies here. And we can actually see how much of the how much of the binding partner, how much of the antibody do we have, how much of the complex do we have, and we can measure the actual amount of them, and then just calculate without much of math, because we just have to take the ratios of the species, the KDs. We're doing that now for like more realistic samples, like larger complexes, which makes it definitely harder, but in principle this is possible with this method, and this is one shot KD, few minutes. So this is antibodies. And this brings me to the end. I want to thank the people involved. So the first part was completely done in Holger's lab with colleagues Ashwin Chari and Jan Kirvis from Holger's lab, and about 70 or 80 collaborators that sent us samples that I don't list all here. And the second part was done mainly by Katharina and Tanya, with a lot of other collaborators from the outside, most importantly Philip and Nick on the APC story, and Nick is also the one who pays Tanya, and lots of other collaborators from the Institute and from all over the world. And I thank you, and I'm happy if you have questions.